No, they came in the front door.
I thought it was like a week It's so nice because sleeping for an extra Oh, you really gonna do this?
Literally my dad's other child.
It's a little less left.
Alright, good morning guys. Go ahead and get started. Quiet down. Alright, I'm going to take attendance real quick. Briley? Here. Brianna? Aiken? Drew? Here. Bria? Here. Henry?
Here.
Anna? Here. Nick? Abby? Hannah? Here. Rosa? Here. Matt?
Here.
Jillian? Here. Mia? Annie? Brianna Hills? Here. Brandon? Sadie? Here. Jessica? William? Marissa? Anna? Here. Sophia? Here. Aubrey? Here. Matthias? Here. Colin?
Here.
Olivia? Olivia P. Braden? Here. Amber? Here. Katie? Here. Ben?
Here.
Ava? Here. Kendall?
Here.
Emily?
Here.
Lauren?
Here.
Jennifer?
Here.
Isabel? Okay, great. So, any questions before we get started on any of the material we went over so far? Has anybody gone to the lecture recordings and watched them or used them at all yet? Okay, cool. So we're going to finish chapter 2 today. I will say at the suggested readings I did remove dark field and the other types of microscopy, electron microscopy, so you can ignore those sections in the book. What's your name?
Brandon.
All right, so we stopped when we discussed simple staining. Because we did that in lab yesterday. How did you guys feel about the simple staining procedure in lab? Okay, cool. So today, we're going to talk about another kind of stain. So a simple stain, basically, it's simple. You use one stain, and you know that it's going to stain those cells that color that you give it. So we used crystal violet, so the cells had that purple color. We're going to also be doing a differential stain. The most famous one is called the Gram stain. Have you guys heard of the Gram staining procedure before a little bit? Okay, so it's actually pretty cool because it's based off of the composition of the cell walls of bacteria. It targets that cell wall structure, and there are two types of bacteria, gram-negative bacteria and gram-positive bacteria, and they differ in their cell wall structure, and so we take advantage of the Gram stain to differentiate between gram-positive and gram-negative cells. That's why it's called a differential stain. So gram-negative bacteria usually will be pink, and gram-positive bacteria will be purple, and we'll talk about how they come to be those colors. So this stain was devised in the 1800s, and basically gram-positive bacteria are purple because they retain the crystal violet stain because gram-positive bacteria have a thicker cell wall that can retain the crystal violet stain. Gram-negative bacteria have a thinner cell wall, so they don't retain the crystal violet stain, and instead they are pink because we give them there's another stain we use that isn't heavy as the purple color. So we'll go through the procedure in a couple different ways just to kind of get you guys to understand this. So here we have gram-positive cells and gram-negative cells. When they are, on a slide, you're not going to be able to see any color. So you fix them to the slide. Here it says add methanol. What did we do yesterday to fix the cells to the slide? Heat fixing. Yeah, we used heat. Good. So at this point, they're clear. If you look at them under the microscope, they're going to be clear. Then this is the Gram stain procedure. You add crystal violet, which is what we used yesterday. And so at this point, the gram-positive and the gram-negative cells are going to be purple because that's the only stain we've added. The second step in this procedure is you add iodine. Iodine binds the stain to the gram-positive cells. So that's just what iodine does. So basically, there's an iodine complex with the crystal violet that allows it to retain the stain. It really locks in there in the gram-positive cells. In gram-negative cells, you can add the iodine. It's not going to wash off the purple stain because iodine is no other color, it's just clear. But because of the cell wall differences, here the purple stain stays reversible. So it's locked in when you add the iodine into the gram-positive cells, but it becomes reversible when you add the iodine to the gram-negative cells. Then you wash with a decolorizer, or ethanol in this case. So because this dye was locked in the gram-positive cells, they stay purple. But in the gram-negative cells, you wash with ethanol, something that's going to remove color. Think of alcohol, acetone, things like that. They remove paint and color. The stain is removed from the gram-negative cells. And then the last step of this Gram stain procedure is you add a counter stain. The counter stain is what we call safranin. It has a pinkish reddish color, and because the gram-negative cells lost the crystal violet, they are going to counter stain with safranin. And the gram-positive cells, they still absorb the safranin, but the safranin is a lighter color than the crystal violet, so the crystal violet kind of takes the lead here. They end up staying crystal violet. So does that make sense so far? Okay. So another way to look at it in terms of the cell wall structure, here you have gram-positive cells, and in the cell wall there is a peptidoglycan layer, which we'll talk about more in this lecture. Gram-negative cells have a very small layer of the peptidoglycan, and so here the crystal violet's added, and it's really being taken up here. The iodine helps the crystal violet lock in, decolorizer, or the ethanol, will release the loosely trapped stain from the gram-negative cell wall because it's not really packed tightly in there because the cell wall is pretty thin. And so that's why these can ultimately stain with the safranin. Okay. So, any questions on that? You'll definitely see more of this as we go on. Okay. So some other stains that we will use in lab, one is the negative stain. Basically, what it does is it colors the background instead of the actual cells to help make things more visible. And then there's the spore stain. Some bacteria and fungi make spores. It's a defense mechanism, it's a survival mechanism, and so their spores will actually do this exact experiment in the lab where you can see the spores are green and the cells are red, so you can see exactly which cells have made spores. Okay, so now that I have diverted a little bit, let's go back and think about the gram stain. What is the correct order of the reagents in the gram stain? Take a second. Who wants to try this? Yeah. B. Crystal violet, iodine, ethanol, or the decolorizer, and safranin. Any questions on that? So it's one of those things you guys should know, the order of the gram stain. Okay. So that's where I'm going to stop for chapter 2, and we'll get started with chapter 3. Okay, so these are the only 3 sections in chapter 3 that you guys should read as supplemental reading. And so we're going to talk about the bacterial cell, its structure, its function. I know you guys have probably already gone through some of this in regular bio 131, but it's really important to kind of give you an overview again. So this is a bacterial cell. You have an outer membrane. It has a cell wall, which is that peptidoglycan layer. There's an inner membrane. There's ribosomes, a nucleoid. Here's like a flagella at the end. So synopsis of the bacterial cell. We're going to talk about cell membrane and transport, and then the cell wall and other layers. So most bacteria share some fundamental traits. They have that thick outer envelope, they have a compact genome, and tightly coordinated cell functions, which allows bacteria to actually be able to replicate by binary fission and just basically make clones of itself over and over and over again quickly. So that's bacteria. They are prokaryotic. Archaea are also prokaryotes. But they have their own unique structures. Eukaryotes have a nucleus, which prokaryotes do not, and eukaryotes have extensive membranous organelles. This should be just a quick review. Okay, so in the early 20th century, the bacterial cell was presumed to be this bag of soup full of floating ribosomes and enzymes. So essentially, everybody thought that bacteria just kind of had, the cell wall, and then you just had this bag of soup inside. And so this is, even up until recently, we're still discovering some new things about bacteria. Modern research shows that the bacterial cell's parts aren't actually as free-floating as we had thought. They're actually pretty ordered. They fit together in an ordered structure, and that structure is flexible and adaptable to changes in the environment. And if you think about how ubiquitous bacteria are, they've been around before we've been around, and they've been able to adapt and survive to many different conditions. And so, there's got to be some sort of order and structure to them. And so they're so small, and we're just now, developing all of these technologies to be able to visualize them in all of their structures. So I really want to hone in on this thought, because even till today, a lot of people still think that bacteria are still kind of this floating bag of soup without this order and this structure. It's really exquisitely organized. Just a general model of a bacteria. So this is a gram-negative bacteria. This is actually E. coli. It's what we used in the lab yesterday. Now, side note, yesterday we stained E. coli with crystal violet, and it was purple, right? If we were to do the gram stain, would it be purple or pink, given that I'm telling you it's gram-negative? Pink, yeah. So yeah, try to keep that in mind for next week. But just a general model of the bacteria, you have an outer membrane, you have the cell wall, it's very thin over here in yellow, and then you have an inner membrane, and then you have, all of this exquisitely organized structures, ribosomes, DNA, in the cytoplasm, and then you have the nucleoid with all of the DNA. There's a flagella, which is used for transport and movement with this rotor or motor over here. Okay. So all cells share some common chemical components. So, they're made of water, essential ions, they're necessary for enzyme function, small organic molecules like lipids and sugars, and then the macromolecules like DNA, RNA, and protein. But the composition of the cell will vary with species, their growth phase, their environmental conditions, but these are the common components that they share. Remember, macromolecules are the polymers of life, so they're definitely going to be a big part of the bacteria. Fun fact, Dr. Fred Neidhart, he basically started this bacterial chemistry field at U of M. And so you don't need to remember any of these numbers for any exam, but from Dr. Neidhart's work and others, the molecules of a bacterial cell basically are broken up like this, where you have about 70% water, 16% protein, 6% RNA, a small percentage of lipids. DNA is only 1%, so just to give you a little perspective. Okay, so the cytoplasm, which is over here, it's contained by a cell membrane, or what we call also a plasma membrane, so that's up here. It's just contained by, having some sort of membrane outside of it. And so the cell membrane is really, really important because it essentially defines the existence of the cell. The cytoplasm has to be contained in order for it to actually be functional, and all of that has to do with the cell membrane. It really defines self from non-self. So the cell membrane helps to distinguish from the actual cell and the external environment. So this is the cell membrane. Can anybody remind me what this is also called, these two layers? Phospholipid bilayer, yep. So the phospholipid bilayer is, really important for allowing things to come into the cell and exchange, things with the outside of the cell. And so you have these hydrophobic fatty acid chains that are inwards, they're pointed inwards, and then the hydrophilic polar heads, the parts that like the water and the external environment, that are out on the outsides. Does anyone have any questions? Does anyone need more of a reminder on the phospholipid bilayer and what it's made up of? Okay. There are other things that you could that are in this image. There are these proteins that are embedded in the cell membrane. These are used for transport in and out of the cell. And so there are also these hoploids. They're stiffening agents that generally people just don't talk about them in general biology, but in this class it's a little more advanced, so we'll talk about some of the other components. And so, yeah, the hoploids are stiffening agents. They help to kind of stiffen the membrane and maintain its stability. When things like hot temperatures would destabilize that environment, the hoploids are kind of that backup to help keep the membrane stiff. And so in terms of content and surface area, there's a lot of protein in these membranes. So half the cell membrane is actually protein. The protein is in the form of these transporters and these membrane embedded proteins. There's a lot over here as well that are embedded. Okay. So membrane proteins have a lot of different functions. They help, with structural support for the cell, help with detecting environmental signals, secreting different, virulence factors if they're trying to keep something away, and communication signals, ion transport, energy storage. So these are some of the functions that these proteins have. Okay, and so the cell membrane, in the context of transporting things across the cell membrane is really a barrier to help keep things that are water soluble and other components within the cytoplasm. So how do things get in? How do nutrients get in? How do we remove toxins from inside the cell? There are specific membrane proteins that selectively allow for the transport of things. So polar molecules and charged molecules aren't able to just freely move in and out of the cell. They need help through these specific protein transporters. And so the two types of transport that we'll talk about, I'm sure you've heard this before, are passive and active transport. So in passive transport, the molecules move along their concentration gradient. It does not require energy. The concentration gradient for passive transport moves from low concentration to high concentration. And then active transport, things will move against their concentration gradient, so that will require energy. Quick review on these things. So just like the phospholipids, the membrane proteins also have their own hydrophilic and hydrophobic regions that help lock the protein into the membrane. So you would assume that, if this part of the membrane is hydrophobic, then the hydrophobic regions are going to be inside. And then these are the hydrophilic areas. And so that all has to do with charges, and so we won't get into all of that. So remember, the phospholipid bilayer is made up of these phospholipid heads and these fatty acid tails. These are the hydrophobic parts of the membrane. And the fatty acids also can vary. They're not all just, the same. They could be either saturated or unsaturated. So if the fatty acid chain is unsaturated, that means that it's more fluid. So here we have an unsaturated example, and you can see in oleic acid, it's definitely more fluid because of this kink over here, like liquid oil. That's why it's able to be liquid. Saturated fatty acids like butter are solid, and so that has to do with their membrane structure and their fatty acids, and so they are more stable and they're not as fluid. So saturated would be more solid at room temperature. But in order for the cells to function, they have to still be fluid to be able to modulate their composition and maintain that fluidity and things that will come in and out of the cell. But you ultimately can't be too fluid as a cell because if it's too fluid, it's going to destabilize the membrane. So fluidity does enhance the function of the membranes. And so some things that contribute to fluidity are if you have these kinks. So here we have a double bond, so you get this kink. And then also the cyclic structure. So if you have a cyclic structure in the actual chemistry of the fatty acid, then that would make it more stable and less fluid. And so just another reminder, there are also these haploids. They add strength to the membrane, so they help stiffen it by limiting the motion of the phospholipid tails. Okay, any questions? Right, so given this information, let's do a quick concept check. So compared to a microbe in our gut, the phospholipid bilayer of a microbe that's living in a hot spring is more likely to have what? So think about a hot spring and the effective temperature on the phospholipid membrane. So something that's living in a hot spring would probably need to have what feature in their lipid bilayer? Yeah.
Are haploids considered lipids?
No, I wouldn't say that they would be. Good question though.
Yeah.
Sorry? E? Yep. So, okay, so let's think about this for a second. That's correct, it's E. So, a microbe living in a hot spring, what does temperature, high temperatures, do to the actual structure? It's going to degrade it, it's going to destabilize it. So a microbe that's living in a really hot environment has to have a really strong membrane. So you would need lipids that will help provide greater membrane stiffness. So I see why you would ask if hopanoids are.
I didn't know if it was like a trick.
It's not, yeah, exactly. It's not, but you would just need some sort of lipids that would stiffen the structure. So lipids providing greater fluidity, you wouldn't want that because that would, increase the fluidity. More unsaturated fatty acids, basically those also will cause more fluidity. You don't want that. And remember, less cyclization means you have more fluidity. So this is the example of cyclization, and then the kinks over here. Okay, does that make sense to everyone? I guess the hopanoids, I guess this part would be considered a lipid, but because of all these OHs, that's why I wasn't sure. But yeah, that wasn't a trick question. A good question. Okay, does this make sense to everyone? And then D, more peptidoglycan, well, we're not talking about the cell wall. We're talking about the phospholipid bilayer, so you can eliminate that one. Does that make sense? Okay, so just trying to orient you guys to the types of questions that I could ask on an exam. May or may not see these questions on an exam, so make sure you take your notes and write the answers down because I don't give you the answers on these. I just give it in class. I won't have a slide with the answer. Okay, so now let's talk a little bit more about the bacterial cell wall. So basically, how do prokaryotes, like bacteria and archaea, protect their cell membrane from dangerous things in the environment? Well, they have at least one structural supporting layer, like an external type of skeleton or an exoskeleton, that's located outside of the cell membrane. And that structural exoskeleton is mostly or most commonly the cell wall. So here we're looking at proteobacteria, it's gram-negative, and you'll notice that in gram-negative cells, they have this thinner peptidoglycan layer like we talked about. There's that inner membrane, this is the cell membrane, but then they also have an outer membrane, whereas gram-positive cells have the peptidoglycan layer, it's really thick, but they only have that one membrane, that one cell membrane on the inside. So that's one of those differences. So the cell wall is really, when it all is linked together, it's this single molecule. It's sometimes also called a sacculus. It provides structural support for the cell. It helps to confer the shape and the rigidity. It helps with things like withstanding turgor pressure. And once it's all linked together, it kind of has this nice cage-like structure. So basically, it's made up of these glycan strands, basically these sugars, N-acetylglucosamine and N-acetylmuramic acid. Those are sometimes abbreviated as NAG and NAM. And so over here we see the peptides are the colorful dots right here. So these are some peptides like alanine, glutamic acid. These are amino acids that help cross-link and connect these glycan strands together. So it's all really interwoven really nicely. So the bacterial cell wall is made up of these glycans, these sugars, NAG and NAM, and then there are also these polymers of amino acids in between that link those structures together. The peptides then can form these cross bridges that connect those parallel strands. So these can also connect to each other and not just to the NAG and NAM. So it's really, fit and woven tightly together. And lastly, we have a transpeptidase. So transpeptidase is an enzyme because it ends in ACE. Almost all enzymes end in ACE. And so that transpeptidase is the last piece that helps click the amino acid groups together that allows for this really rigid cell wall for the bacteria. So it's involved in cross-linking the peptidoglycan layer. So glycans are sugars, peptides are amino acids. So the cell wall is made up of the peptidoglycan layer, amino acids, and sugars. It's in the name. You should know that. And so over here we have the phospholipid bilayer, and that's protected by the cell wall. So this is the peptidoglycan layer. You zoom in and you see it's made up of peptides, and glycans. You also have the transpeptidase enzyme that clicks things together. I actually created this image and I'm proud of it. Any questions so far? Okay. So another, way to show you the same thing, you will have to memorize the actual structure. I'm just kidding. This is not biochem, you don't need to remember any of this, but you should, understand what is involved. So this is just an FYI slide. And so the reason I want to show you this is because this peptidoglycan structure is really important, and there are a lot of antibiotics that target this specific process. It's this rigidity of the cell wall and this peptidoglycan structure. So has anybody here taken amoxicillin or penicillin? Probably every single one of you. Yeah. So amoxicillin and penicillin, it's a penicillin class antibiotic, that actually targets the peptidoglycan layer or the cell wall of bacteria. So because peptidoglycan, or the cell wall, is unique to bacteria, we don't have cell walls. So we can use it as a target if we have a bacterial infection. So we can create antibiotics that will target the cell wall of bacteria, but it won't harm us because we don't have a cell wall. So it makes an excellent target for antibiotics. So the transpeptidase enzyme that cross-links the peptides at the very end is actually the target of penicillin. So penicillin, it's a beta-lactam antibiotic. You don't need to know that for this class, but beta-lactam antibiotics basically are bactericidal agents that interrupt bacterial cell wall formation by binding to the transpeptidase. They're called beta-lactam antibiotics because they have a beta-lactam ring, which is over here. You don't need to know that, but I like to have all of the information for you guys. So this is a beta-lactam ring. It kind of looks like a house, sort of. And so the way that this works is the beta-lactam ring of the antibiotic will try to bind this transpeptidase, and it disrupts the cross-linking of the peptidoglycan layer. Then water will rush into the cell, it will lyse, and it will burst because the cell is now not able to be rigid and compact because there's no cross-linking. And so usually that will allow, things from the outside of the environment to rush in, like water, and then the cell will burst, and then the bacteria gets killed. So that's how a lot of these antibiotics work, these penicillin class or beta-lactam antibiotics. But of course, it's really important to remember that the widespread use of antibiotics and the misuse of antibiotics will select for these evolutionary resistant strains. And antibiotic resistance is clearly, something we're still fighting against. And not all antibiotics will target peptidoglycan synthesis, but just these ones do, the penicillin classes. Any questions on that? Does that make sense? How the transpeptidase enzyme clicks things into place, and so those antibiotics will bind to the transpeptidase enzyme and interrupt that clicking, cross-linking. Okay. So the cell envelope of bacteria, there's some bacteria that have an additional envelope layer. And so we discussed the inner membrane, the cell wall, both are components of this cell envelope. But the envelope composition really helps to define whether a cell is gram-positive or gram-negative. So, I really like this image. So over here we have a gram-positive cell. This is, a microscope image, but if you kind of zoom into what's in here, you have the cell membrane with the membrane proteins, and then you have, this is the peptidoglycan layers, the NAG and NAM that's linked together. You see it's very thick in gram-positive cells. And then there are some other things and chains in this S layer over here that actually we recently discovered was there. And so, this is just what makes up the top part of it, but there's no additional membrane. There's no additional membrane like in gram-negative cells. In gram-negative cells, you have the inner membrane, a very thin peptidoglycan layer, and then an outer membrane to help with the structure and the rigidity. And the outer membrane is connected to the peptidoglycan layer in gram-negative cells by these lipoproteins, so you guys should know that. The peptidoglycan layer has to connect somehow to the membrane. There are no lipoproteins here because lipoproteins will connect to another membrane and there's no other membrane. And so in gram-positive cells, the cell wall is threaded by what we call teichoic acids. So I really want you guys to know this because this is an important step in the gram stain. Remember when I said that the iodine helps to lock in the crystal violet in gram-positive cells? Well, that's because the iodine interacts with these teichoic acids, and so that's what helps the crystal violet stain lock into the cell. So, summary, multiple layers of peptidoglycan in gram-positive cells. They're threaded by these teichoic acids. And then there is a capsule, so it's kind of literally looks like a capsule around the cells that are made of more polysaccharides and glycoproteins. These usually, they're slippery and they help with preventing phagocytosis and keeping the cell from drying out. So phagocytosis is when an immune cell engulfs a bacterial cell and tries to kill it. And so this capsule helps give it some more protection. So this capsule is also found in some gram-negative cells, not all. And so, yeah, this is dark field microscopy or negative staining. Remember, negative staining helps to stain the background so you can see the capsule better. Gram-negative cell envelope, thin peptidoglycan layer. There's this outer membrane that will confer additional defensive abilities. And then another thing that I want to mention is that there's this outward facing leaflet that is really to, so this LPS layer, it's made up of lipopolysaccharides or LPS, and some porins. And then there's this inner facing leaflet that has like lipoprotein. So that's this part that connects the peptidoglycan layer to the outer membrane. So gram-negative cells have this LPS layer, these lipoproteins, and so some other important things to remember. And then you'll notice that there's this S layer up here, and it's actually in both gram-positive and gram-negative cell walls. This is just an additional protective layer that's commonly found in bacteria. It's a protein coat. It's really crystalline, so it's a thick, protein. It helps to contribute to cell shape and protect the cell from osmotic stressors. So it's rigid, but it's still porous enough to allow things to pass through. Okay. Questions? Okay, quick concept check. Which of the following is not a component of peptidoglycan? Yeah. C, lipopolysaccharide. The LPS is on the outer leaflet of the outer membrane of gram-negative cells, and it's not part of the cell wall. So NAG and NAM are the two glycans, and then you have amino acids and peptide crosslinks in the peptidoglycan cell wall. So before you guys go, we have our first worksheet assignment that you guys have to complete. It's going to be due on Friday. You have to bring it in completed. And so as a class, we'll go through it together. Yeah. You can either print it out and handwrite on it, or you could have your laptops out and have it filled out on your laptop. It doesn't matter to me.
I know like one session's something about like draw.
Yeah, so you might want to print out that part, yeah. Any other questions? Yeah.
What's the answer to the concept?
C.
I'm just gonna the sheet.
Yeah. Completion of participation,
Unless there's like
We're not gonna be here Friday because we have summer night for him.
Yeah.
So
Remind me your name. Sorry, I need last name because
I believe you back.
it's like a delivered to the
That's where I'm trying to actually work.
I usually do
And then either sheet and just like mail it to you.
I was gonna say, if you're gonna be here, email me the worksheet just so I can see that you've done it, and then we'll go through it on Friday. You can watch it.
Thank you. Perfect, thank you. I'm gonna be taking some. I'm gonna just get there early. I can get.